Hello and welcome to another episode of Katie the Science Lady. I'm Mrs. Jacobson and today's topic is mutations. So let's learn together. Okay, today we are talking about genetic mutations and how they ultimately can come to affect our whole entire body because they're affecting the proteins that we make in our cells. Now, genetic mutations are a change in the nucleotide sequence of a DNA molecule. And what this means is it's an error or an alteration in your DNA bases. So in those A's, T's, C's, and G's, you're having a change at the DNA level. Some of these do not change our final protein, and these are called silent mutations because while there may be a change in the DNA, if we don't see a change in the amino acid or the protein, the effect is null. There's no effect at all. It's silent. If our final protein is affected, we call this expressed. And all expressed means is that our protein at the end is different. This doesn't mean that it's a bad thing or a good thing necessarily. It just means that it's different. It's just saying that it's going to have an effect. It doesn't say good or bad. Now, the key thing to remember is that mutations only happen to DNA. We don't have mutations happening at our mRNA level. They occur at the DNA level. So typically, most genetic mutations are passed on. Well, all genetic mutations are passed on from your parents to you, and they occur before you're born. So the mutations are already present when you start to develop. If you gather mutations as you're living, so let's say somebody decides to start smoking cigarettes and they have all those carcinogens that are affecting their DNA and their cells, specific cells may have mutations due to that carcinogen found in the cigarettes. And remember, those small mutations in our cells can lead to cancer, but we're not talking about those mutations today. Today, we're talking about the mutations that you're born with, genetic mutations. So this is a question I get all the time. Aren't they bad for you? Aren't mutations a bad thing? Most of my students come in thinking that the word mutation is really bad. I do have a couple of students that come in and go, hang on guys, the X-Men. And they get really excited because they think we're going to learn about the X-Men or Teenage Mutant Ninja Turtles or something like that. It's not as exciting as all of that, even though I do love the Ninja Turtles. But today I'm here to tell you that mutations are necessarily overall a good thing. They create genetic variation or they create variety between members of the same species. Mutations are why we don't all look the same. We have differences in our DNA that allow us to have different hair color, eye color, heights, different features in our bodies. And overall, this is a good thing. If your mutation is favorable, which means it gives you an advantage and helps you survive, then that's a good thing. So it's not really that it's helpful or harmful. They can be both at the same time. But if it helps you survive and reproduce, it's helpful. If it doesn't, it's harmful. And we call these less favorable mutations. And as it says on the screen, you can see it says they are removed from the gene pool. Typically, that means the organism does not survive, or they don't survive long enough to reproduce. So in that case, it would be harmful. But again, this just depends on the environment we live in. Many mutations nowadays aren't harmful, where they may have been a couple hundred years ago, because we have medicines and therapies and surgery that can help fix some of these mutations or mitigate their effects and make them not so harmful. So it just depends. And that's kind of the tricky part about mutations is they're not necessarily good or bad. They could be completely neutral, but it depends. Now we have a couple different types of mutations. The first I'm going to talk about today are point mutations. And it is what it sounds like. It's one single DNA base or nucleotide that is swapped for another. So I think of it as like a substitution, like if you're watching soccer or basketball or even hockey, any sport really. When you have a substitution, you swap out one player for another player. They just switch. That's what a point mutation is. You're taking two bases and you're swapping them for the next. So an A could become a C, a G could become a T. It just depends. But it's one single nucleotide changing into another one. So if we look at an example here, we can have our DNA that looks like this. We have UCC, and again, this is showing you mRNA, and that will become serine. If we have a mutation that changes that C to an A, and we have UCA now, we could still have serine. So would that be expressed or would that be silent? It'd be silent. There's no change in the amino acid, so we call that a silent mutation. Okay. Now let's take another example, CAU. That's for histidine, the amino acid histidine. If that changes, that A changes to a G, and we have a mutation there, a point mutation, it now becomes arginine. So this would be expressed. We have a change in the amino acid sequence. That means our protein is going to change. It's not going to be the same sequence, so it is called expressed. The third type I want to talk to you about here is if we have the example of UAU, this is tyrosine, the amino acid tyrosine. If it switches to UAA and that last U in the amino acid or in the mRNA sequence changes to an A, we get a stop codon or a stop amino acid. It's not really an amino acid, it's just the stopping point. If this happens, it's expressed because whatever our protein was, it's now stopped. It's the ending. Now, if we have a couple other point mutation examples that I want to talk about. The two that you may have heard of or have seen in the news most likely are sickle cell anemia. This is a point mutation. So we look here and we see I have normal red blood cells. Sickle cell anemia attacks the red blood cells, and particularly their shape. It's going to deal with the hemoglobin protein, which is what carries the oxygen to your body. The oxygen fuses to the hemoglobin or binds to it. So if we have our DNA here, we have GAG is our normal DNA. When it mutates, it becomes GTG. So the A swaps with the T. This changes from glutamic acid to valine. When that happens, the entire protein shifts and it creates these sickled cells. So our red blood cells normally are this nice disc shape, and that lets them get a lot of oxygen into them because our hemoglobin protein needs the oxygen to get it to our body cells. When the cell sickles, like curves, it can't carry as much oxygen. The hemoglobin is not the same protein, so it can't carry as much oxygen, so it has trouble in our bodies, and it can cause some other issues as well. A second point mutation example is lactose intolerance. It's kind of crazy to think that it's just one little tiny letter base that changes to create lactose intolerance, but that's what it is. We see that here we had a C, and it gets switched to a T, and that's all that changes. You have one small mutation that changes the entire protein and makes you unable to process lactose, or the sugar that's found in milk and dairy products. There are a couple other types of mutations I want to talk about. These are not point mutations. Please keep that in mind. These are called frameshift mutations. And the first type I want to talk about are insertions, and they're exactly what it sounds like. You're inserting a base or more bases where they don't belong. So it's one or more new nucleotides. It can be more than one, but it can also just be one. And this causes a shift in our codons. Remember, a codon is a set of 3 bases. So if you went ahead and added an extra base anywhere in here, you're going to have now 4 bases, and you'll have to shift where the codon was. So codons don't get to stay together. If something gets inserted in the middle of a codon, it's going to shift everything over. And it's going to result in a completely different protein strand. Because now, once you insert, let's say here we've got a G that got inserted. Now we have CCG instead of CCA. That's a different amino acid. Now we've got, instead of ACT, you have AAC. It changes all the way down the line. So it has a huge ripple effect on our entire protein. Deletions are, again, frameshift mutations in the same way. Instead of adding a base, you're taking away a base. It's like you click that escape button on your computer and you just took away that last letter. This can, again, be more than one nucleotide, but you're removing it from the sequence. And again, it causes that reading shift. So if we see here, instead of having CCG as a codon, AAC as a codon, TCG as a codon, our first 2 remain the same, CCG, AAC, but now we don't have TCG, we have CGA. So you're going to change everything after that point. So you'll keep some of the protein the same, but then after your mutation, that deletion, everything will be changed afterwards. These are irreversible mutations. Because you've taken something away, it cannot be fixed. Sometimes a point mutation can be fixed by your cells. They can, again, not a genetic mutation, but if it's an acquired mutation, one that's from UV rays or carcinogens or something like that, your body and your polymerases can say, hey, that's a mistake, let's fix it. But if it's a deletion, it's gone. It's not coming back, and it's not possible to get it back. So these frameshift mutations are huge. They can cause really big problems, and some of these mutations can result in death because you are changing the entire protein, not just a small piece of it. Again, we're talking about insertions or deletions here, and it causes that drastic change. So one of the best ways I've ever seen this done is with sentences. If we think of our codons as little words that mean something, because remember they code for an amino acid, so they have a meaning. If we change those amino acids, it's like changing the words in a sentence. It's going to change what it looks like. So if I took the fat cat ate the rat, now if I just changed the R here to a B, did a point mutation, it would be the fat cat ate the bat, which is a completely different story. I have questions about that. How did this bat get caught by a cat? Why was the cat next to the bat? I have lots of questions about it. Completely different sentence, completely different meaning. However, if we do a frameshift mutation, the sentence won't even make sense anymore. And I'll show you what I mean. If we add in or insert a letter A here, it went from the fat cat ate the rat to the fat cat ateth or at. It's not English anymore. It's not a sentence anymore. It makes no sense. So everything after that insertion point is changed. Let's take a look at deletion as well. Now we have, instead of the fat cat ate the rat, we have taken out that A. So that first A here in 8, we've taken it out or deleted it. So now we have the fat cat teter at. Again, doesn't make sense. So we have that issue where if you are doing a frameshift mutation, you're adding or deleting something, the whole protein sentence or that a whole amino acid DNA sentence is completely messed up and you're not going to get the same result. Again, we deleted that A from there.
Let's recap what we've learned about mutations so far. Mutations are changes in the DNA sequence of a gene and can be ultimately helpful, harmful, or neutral and have no effect at all. Mutations can be expressed, which means that the DNA change is also shown in the amino acid sequence, changing the shape of the protein overall. Or they can be silent, and this means that the DNA change is not seen in the amino acid sequence, and it stays the same, meaning the protein also stays the same. We also need to clarify a few things. Genetic mutations occur before birth and are passed from parent to offspring, while acquired or accumulated mutations occur in single cells and are usually due to things like environmental stimuli like UV rays and can lead to disruptions in the cell cycle and cancer. Thank you for watching. Please like, subscribe, and comment for more biology content. And until next time, I hope you had fun, I hope you learned something, and I'll see you later.